Hello everyone, and thanks Lucy again for this opportunity to share some of my recent work on cyanobacterial calcification. And as Lucy mentioned, I'm gonna talk on spectroscopic insights on intracellular ACC formation in cyanobacteria. Before I go any further in my presentation, I just wanted to say a huge thanks to the team which was involved in making this work happen. It's a vibrant team of people, including the research group Biomin, which I'm currently hosted at as a postdoc, headed by Karim Benzara, and various other groups, all at Sorbonne University. As the title states, my talk is going to revolve around two main actors, cyanobacteria and carbonates. Many of you in the audience probably know that cyanobacteria are the most ancient life form on the earth. They are unicellular photosynthetic bacteria biomineralizing calcium carbonate as a byproduct of photosynthesis. This process has multiple implications for geobiological application in forms of signatures of life in ancient microbolites, as well as modern analogs of ancient life form in form of stromatolites. But they are also have numerous engineering application in terms of CO2 sequestration, as well as synthesizing bio-inspired products. However, this paradigm that cyanobacteria were capable of inducing extracellular calcification was challenged since the discovery of cyanobacteria Bleomargarita lithophora, which is shown here, right here with on an SEM image. This cyanobacteria was shown to form intracellular amorphous calcium carbonate. And these carbonates are here, if you can see it, with the bright sphere, which are typically in range of 100 to 300 nanometer in diameter. Since then, several cyanobacteria, not only from diverse taxa, but also diverse environments, have been shown to form intracellular ACC. Some examples have shown here in the STEM EDX images. And for some of you who are seeing these images for the first time, just a word of caution. So these are just the STEM images, and here is a chemical map where the green sphere basically shows calcium hotspots, whereas the red sphere shows a phosphorus hotspots. And the calcium hotspots are interpreted as ACC. Some other examples of this diversity also includes multicellular cyanobacteria, bloom-forming, sometimes toxic cyanobacterium such as microcystis, as well as some marine cyanobacterium strains. Currently, the exact function of ACC remains ambiguous, but some current hypotheses include its role as a pH buffer, as well as a storage for inorganic carbon and/or calcium. The repertoire of intracellular amorphous calcium carbonate forming bacteria is not just limited to cyanobacteria. There are a wide variety of prokaryotes which have been increasingly being reported to form ACC. Two notable examples here is a sulfobacteria Acromatium oxaliferum, which was recently shown to possess forming these huge amorphous calcium carbonate inclusions, and as well as a magnetotectic bacteria, which was affiliated to alpha proteobacteria, was also shown to possess the ability to form ACC. However, despite the widespread reports of ACC forming prokaryotes, searching for ACC forming bacteria in an environmental sample is not straightforward as one would imagine. Prokaryotic ACC can be lost and are transformed upon typical sample preparation protocols. So for example, many of you in the audience who are familiar with microscopic techniques on biological cells, a common approach is to use fixatives. But it was shown that traditional fixatives can transform ACC inclusions by Lee et al. And as well as in some of my work. Moreover, sometimes instrument setting itself could transform ACC and induce in situ transformation of ACCO to a crystalline phase, as shown by Benzara et al. In 2020. So even a partial loss of ACC may become problematic for samples with limited amount of ACC forming bacteria. And to give you a context on the amount of samples of ACC forming bacteria in an environmental sample could be, Bradley et al. And Amouchial et al. Showed that up to 10 to 15% of the total microbial population can be attributed to ACC forming cyanobacteria, which is not a lot. Although current instrumentation techniques such as SEM, STEM have been incredibly successful in detecting ACC in bacteria, these techniques are powerful in providing single cell scale information. But their use is time consuming when the question is simply to know a preliminary confirmation of the presence or absence of ACC in a sample. Overall, there's a need for the use of techniques allowing efficient detection of ACC in these complexes samples with minimal sample prep. So here in the first part of my talk, I will present an approach using Fourier transformation infrared spectroscopy to answer this question. FTIR is one of the most commonly used form of vibrational spectroscopy, and it's capable of rapidly detecting calcium carbonate polymorphs in situ in a non-destructive manner and without complex sample preparation, making it an ideal approach for detection of ACC in prokaryotes due to the aforementioned reasons. Just so that we are all on the same page, I'm sure people already are aware of FTIR technique. Here I've presented a quite simplified working principle of FTIR. Basically, it involves irradiating a sample with IR radiation and recording the absorbance of IR radiation by the sample. At the end of the analysis, one gets the output, which is shown here as an example, where the x-axis is the wave number, or you can call it as a frequency, and the y-axis is absorbance, which corresponds to the amount of IR radiation absorbed at different wave number. The spectral features, that is the peaks, which you observe here, are characteristic of the type of molecules which are present and its chemical environment. And thus, that's the reason why FTIR can be a very powerful fingerprinting technique. So now we all know the basics of FTIR spectroscopy. Going back to our original question of how can we use this technique to detect in situ ACC forming bacteria. And to do so, we propose a development of an IR spectral probe that could serve as a reliable marker of ACC forming bacteria. In order to develop such a probe, the first step involves knowing what are the characteristic IR vibrations of pure amorphous calcium carbonate. So here I'm showing two spectra. One spectra is the ACC spectra recorded in midIR region, which corresponds to frequency between 4000 to 400. And on the right is the farIR region ACC spectra, which corresponds to frequency region between 600 to 50. The arrows which you see here marked on the figure, they denote the frequency of the location of the peaks. And each peak is associated with a particular type of molecular vibration of the carbonate ion which is present in ACC. So having known the location and peak shape of the characteristic ACC peaks, now the next step involves in analyzing which of these characteristic peaks of ACC remains preserved in a known ACC forming cyanobacteria. So to do so, we compared IR spectra of ACC forming strain, which is here shown in red, with that of a pure reference amorphous calcium carbonate, which I showed you in the previous slide. What we observe is that in the mid-IR spectra of the ACC forming strain, the majority of the ACC vibrations, which are again shown here by the arrows, were not as well defined as in the pure ACC in comparison to the, I'm sorry, the characteristic vibrations of ACC were not as well defined in the ACC forming cyanobacteria when compared to the reference ACC compound. In fact, all the characteristic peaks of ACC in the region above 900 centimeter, which is between 1800 to 1000, were in fact masked by vibrations from the background biomolecules such as proteins, lipids, fatty acids, so forth, which are commonly present in a cyanobacteria or for that matter, any prokaryote. So these frequency of characteristic peaks were rendered useless when you want to detect ACC signature or a marker in ACC forming cyanobacteria. However, what we did find that we could clearly observe that characteristic band of ACC at 860 centimeter minus 1, which is right here, shown by the black arrow, was preserved in an ACC forming cyanobacteria, shown here in red and labeled by the number 11. More so, we know that this band was absent in a non-ACC forming cyanobacteria, which is shown here in green, suggesting that the presence of band 11 is due to the presence of ACC in the cyanobacteria and rather than any non-ACC component. Together, what it suggests is that we have a potential spectral marker which is located at 860 centimeter minus 1, which we can use further to probe for the presence or absence of ACC in a cyanobacteria and probably in other prokaryotes. Consistent with the mid-IR observations, we also investigated the far infrared region looking for a spectral marker. In the FIR spectra of ACC forming cyanobacteria showed a broad but strong peak around 300 centimeter minus 1. Excuse me. This peak, which is labeled here with letter A, was absent in a non-ACC forming cyanobacteria. The position of this peak coincides with that of a broad peak observed in the FIR spectra of a synthetic ACC, suggesting again that this peak might be due to ACC rather than any non-ACC component. Thus, this offers a second spectral marker of ACC for the purpose of finding them in the environment. Furthermore, we applied the two identified spectral probes for detecting ACC forming cyanobacteria to some unknown samples of cyanobacteria. Using these two spectral probes, we were able to screen 4 microcyst strains whose ACC forming capabilities were unknown. So here I'm showing just a subset or a small region of the IR spectra of microcyst strains. We analyzed about 8 of these strains, and the solid line here is a guideline for 860 centimeter minus 1 spectral marker for ACC. The prediction of the ACC forming capabilities by microcyst strain was later confirmed as well by SCM EDX and molecular approaches, and thus was consistent with FTIR observations. So altogether, this approach showed that the V2 band, which is what I call this 860 centimeter minus 1, and the broad peak around at 300 centimeter minus 1, both of which are characteristic of ACC, were not only free from interference from any non-ACC components such as the proteins or polysaccharides and so forth, but they were also only present when a cyanobacteria contained ACC. And therefore, we argue or propose that these both spectral features of ACC are promising probes for detecting cyanobacteria possessing the ability to form ACC. So far, I have shown you how IR spectroscopy could serve as a practical tool to identify ACC forming prokaryotes in the environment. In the remainder of the talk, I will shift gears a bit to shed some light on the fundamental aspects of ACC formation and its stability in cyanobacteria. So ACC is widely studied in skeleton-forming organisms, where ACC is precipitated to be transformed into crystalline calcium carbonate polymorphs. However, in cyanobacteria, this metastable ACC phase remains amorphous for the life of the cyanobacterial cell. And therefore, it raises a question, how are these transient phases stabilized? Previous work from Benzara et al. Group have shown evidence of a specific cellular compartment surrounding ACC using cryo-EM studies and proposed that this cellular compartment may play a role in stabilizing the ACC which is present in these cyanobacteria. In my work, I'm interested in knowing what mechanisms are involved within this cellular compartment that may stabilize ACC. Is it driven by more of a confinement strategy, which has been proposed as a way of stabilization of short-term ACC in many of eukaryotes? Or it is driven by additives such as organics or inorganic additives such as magnesium, which has also been proposed by many studies to play a role in stabilization of ACC in eukaryotes. And lastly, I'm also interested in knowing what is the chemical environment surrounding ACC. Is this a hydrated shell or is it an organic matrix? This is something which is unknown, and that's part of my work which to dig deeper into this question. So to answer these questions, here I use a solid state NMR technique, which is a powerful and robust technique, and yet another spectroscopic technique to determine structural and molecular functionality in situ. ACC is very easily differentiated from other calcium carbonate polymorphs such as calcite, veterite, or aragonite because of it has a very characteristic broad peak, which is shown here, and it's located around 169 ppm chemical shift. So this feature is what we're going to use to detect ACC in our samples using NMR. So in our system, the way we approach this this study was that we use solid state NMR to probe intact cells of ACC forming cyanobacteria so that we don't induce any transformation of ACC during sample prep. Accordingly, so cells cultures were labeled with the NMR active nucleotides. Here it was 13C. And the cultures were grown to a high density to produce large amount of biomass. And this biomass was dried and packed into an NMR rotor for analysis. So on this slide, I'm showing a 13C cross-polarization NMR spectra for ACC forming and non-ACC forming strain. So the ACC forming strain is in red and a non-ACC forming strain is in blue. I know this slide is a little bit dense, but if you can just focus on this gray box here, which corresponds to the region where you would see any characteristic bands of ACC appearing. So if we zoom into this region, the spectra of ACC forming and non-forming strain almost look identical, except that there is an additional peak at 169 ppm present only in ACC forming cyanobacteria. This peak at 169 ppm matched in shape and location to reference ACC spectra, and when you compare it with other carbonate polymorphs, it was not matching any of the peak shape or the location. Moreover, by the same reasoning, we didn't find any crystalline polymorph peaks in the NMR spectra of ACC forming cyanobacteria, suggesting that the sample preparation method used in our study didn't induce any transformation of ACC. Altogether, this exercise validated, first of all, our experimental protocol that we were able to detect ACC in its native state and in the intact cells. But now going back to the more of a fundamental question about what is the chemical environment around that specific cellular compartment which I showed earlier. So through our cross-polarization experiment, we were also detected that ACC in cyanobacteria was in proximity to a protonated species. The chemical environment of this proton was revealed from a 2D proton 13C NMR, which showed a single correlation spot for ACC corresponding to a proton shift of 5 ppm, which usually corresponds to water. All in all, for people who are not familiar with the 2D NMR plots like this, all this is trying to say is that ACC in cyanobacteria was found to be hydrated. And this is not a surprising revelation because we know that hydration allows or plays a role in stabilization of ACC, but it is for the first time we have shown that even in cyanobacteria, or for that matter, prokaryotes, ACC was hydrated. In our preliminary data, we didn't find any traces of any organic environment in the vicinity of ACC. And this was interesting because usually in eukaryotes, you would see a signature of an organic matrix in the vicinity of ACC, which has been hypothesized to play a role in its stabilization. So having validated our method and established that ACC is hydrated, next we focused on carbon-phosphorus interactions using 13C, 31P REDOR NMR spectra. We focused on phosphorus for one particular reason and that is phosphorus has been widely implicated as an additive to stabilize ACC. And these studies have largely focused on eukaryotes primarily, and also some of the abiotic synthesis of ACC. So here on the left is the full spectra of ACC forming cyanobacteria. The top spectra is a reference 13C spectra, which shows all carbon species which are present in a cyanobacterial cell. Whereas the bottom spectra is the difference spectra that shows only those carbon species which are present in the proximity of phosphorus. What I want you to focus on is again this black rectangle, which represents the carbonate region in an NMR spectra. When we zoom in, based on the different spectra, we find evidence of phosphorus presence of phosphorus in vicinity of ACC, suggesting it's possible that phosphorus also plays a role in stabilizing ACC in cyanobacteria. The nature of the phosphorus species remained to be determined. A plausible candidate could be a polyphosphate inclusions, which are also present routinely in ACC forming cyanobacteria as well as in general in prokaryotes. But we need to do a further studies in order to constrain the speciation of these phosphorus moieties in vicinity of ACC. And with that, I want to point out some take-home messages that ACC environment was in our study was successfully probed in intact cyanobacteria using solid-state NMR, and that revealed that ACC is not only hydrated but also presence of phosphorus and its role in ACC stabilization. And to connect these findings to a more of a broader picture, earlier in my talk, I mentioned about the various types of prokaryotes and eukaryotes from diverse environments are found to form stable ACC. Hopefully, I've at least convinced you a little that owing to the biological simplicity and ease of culturing of cyanobacteria forming ACC, ACC forming cyanobacteria could be used as a model system to better understand the mechanisms of ACC stabilization, as well as understanding their occurrence and formation in complex environmental system. With that, I'm going to stop here so that there's plenty of room for questions. So yeah, I'll hand it to you, Lucy.
Yeah, thanks a lot. Oh, so we've got already a question from Martin, and thanks, by the way, for the very clear presentation.
Thank you.
Martin, yeah, can you activate your microphone? Yeah, should be all right. Martine, ouais. Je sais pas pourquoi Martine, mais j'ai pas accès à désactiver moi.
Attends, voilà, ça y est. Attends, hein?
Yeah, it's all right.
Sorry. So I wanted to know what is the role of this carbonate in this bacteria?
Yeah,
because it is so different. I suppose the environment are very different, and in the bacteria are all the time accumulating this carbonate, or there are particular reason, or there are essential for some steps of their development.
Yeah. So that's an excellent question, and so I'm going to give you a long answer to it because it's not straightforward, and partly it's not known. So as you probably know that whether it's cyanobacteria forming ACC or not, calcium is an essential element, and it's needed for the functioning of various essential functions of cyanobacteria. What happens in the ACC forming cyanobacteria, where it was first at least identified, this was in an alkaline lake in Mexico. And what it was observed is that the extracellular calcium concentration was way below to the state that there was no reason to expect any oversaturation or precipitation of a calcium carbonate phase. Second of all, when you take that extracellular chemistry, you would not see any precipitation in a laboratory culture, for example. Third of all, when you have almost 6 order low concentration of calcium, you still see these cyanobacteria forming ACC. So there was no dependency whether you have low or high calcium. The ACC formation was always active. The only thing which was seen there was this paper by Weaver et al., what was seen that the calcium demand for ACC forming cyanobacteria was quite high when you compare with that of let's say, a non-ACC forming bacteria. And in fact, when you try to culture them, let's say in a media which has no calcium, they eventually don't grow. And if you keep decreasing calcium concentration, their growth curves also, their growth reduces, or it's almost inhibited. So that's the relationship to calcium. Now, when you ask about the function, and that is something we have no clue why calcium carbonate inclusions are formed. As I said in the beginning, there are plenty of hypotheses when this was first discovered in Gliomargarita lithophora. So first was that it could be a storage form of inorganic carbon because it's at the end it's calcium carbonate, and it's very easy to dissolve or ACC when it rather than a crystalline calcium carbonate. The other option was as a ballast to allow cyanobacteria to maintain a benthic versus a planktonic lifestyle. But given that the cyanobacteria are usually in the photic zone, this hypothesis might be applicable for some other kinds of prokaryotes, but probably not for cyanobacteria. The other hypothesis is that, which is the most prominent, is that it acts as an intracellular pH buffer. By which I mean is that we know there's a list of different kinds of pH buffers which are maintained in the cyanobacteria, because all the cyanobacteria, and for that matter prokaryotes, try to maintain or regulate intracellular pH within a range of 6.5 to 7. So we suspect that ACC precipitation could be linked as a means to control or regulate intracellular pH by these cyanobacteria. The question is, why do they do it, or why is pH so difficult to regulate in these cyanobacteria? For that, we don't know. For that, we have suspected that it could be linked to the transport mechanisms, what transporters they have for alkaline earth elements.
It was one of the hypotheses, one of the things that crossed my mind.
Yeah.
That in fact, is it possible that when the ACC is made, they just get rid of it? They secrete it. Do they secrete it?
No, so they don't.
Or it stays inside the bug?
Yeah. So once the calcium is inside, it stays inside. It doesn't get out. So it's only an influx. There's no efflux of calcium. So the ACC, once they are formed, they remain inside. We did see in some strains over the growth stage of the cell that you would see that the ACC might disappear from the cell, and that's been suspected because maybe calcium is needed as they are growing at a later stage and there's no more calcium in the growth media. But for many of the strains, ACC just stays intact inside the cell for the lifetime. Yeah. So I don't know if it answers fully your question, but
yeah. Okay. Thank you very much.
So thanks a lot. We've got now a question from Nadine.
Good morning, everybody. Thank you very much. It was really interesting. Maybe you mentioned it, but I miss it. I have two questions. I would like to know first if you have tried to push the crystallization at the to see how this amorphous calcium carbonate may crystallize and see if there is some different kind of polymorph or it's really stable. And the second point is concerning the NMR. Did you try to do to perform solid state NMR, but with on the phosphorus environment to see if it's really a phosphorus that is related to organic compounds or inorganic compounds?
Okay.
Thank you very much.
Thanks, Nadine. So for the first part about trying to crystallize intracellular ACC, we have tried a couple approaches. In the first approach, we actually tried to extract it outside of the cell and induce a controlled crystallization. However, the problem with that approach was that since the environment inside the cell was undersaturated with respect to carbonate phase, as soon as they were extracted outside, whether it was let's say in a chloroform or an organic solvent or in any oversaturated with respect to calcium phases, we had no control over what phase it would form. The second approach we tried was actually we tried doing a localized heating via laser, so Raman spectroscopy, because when you increase the Raman laser power, you can actually induce transformation of ACC to calcite. And that's what actually we saw what happened in acromatium, where you increase the laser power to 100%, you instantaneously transform ACC into calcite. But that said, we don't know if when such a transformation happened, how much of that envelope, which I spoke about, the cellular compartment, how much of that would remain preserved. So it's an interesting idea to explore a little more about how this gets stabilized and how we can work more easily, but we haven't figured out a way to better control its crystallization.
Thank you.
Yeah, and then for the second part, for the NMR aspects, so that has been very interesting because we got super excited when we saw the radar spectra that indicated that there might be some phosphorus, and we wanted to look into the different species, and we tested different kinds of sequences to probe into like whether it's an organic environment. But what turned out that we didn't get any signal when we tried, let's say, sequences like REDOR or headcore, for example, which have been known to elucidate the phosphorus environments in vicinity of ACC. we tried it for like on our sample, we just couldn't get any signal. And we collaborated with some other people from LCMCP, and they did some modeling on these interactions between phosphorus and carbon, and it turned out that probably the recoupling between phosphorus and carbon interaction was insanely weak, and it would be very difficult to observe that interaction in a more of a reasonable time frame. So that's where it is, all the phosphorus-carbon interaction story for the moment.
Thank you very much.
Yeah, you're welcome.
All right, so is there, yeah. Oh, Martin, one more question for sure. Yeah, Martin, you can ask your question, no problem.
Okay. I wanted to know if it is possible to have some of the cyanobacteria to look at it because I have a microscope and I am analyzing if it's possible to get some of it, just to see what I can see with my microscope.
Sure. I'm definitely happy to connect you with Karim because he's the head for the team, and I'm sure we can arrange some sort of arrangement.
Yeah, we have already some relationship with your institute.
Okay.
Because, with Jacques Chaumier.
Okay. Yeah, I will definitely send you an email CCing and connect you with Karim, and you can discuss the possibility with him. I think that's the easiest.
Okay, thank you.
You're welcome.
All right, so I will have a very naive question.
Go ahead.
So is there already any estimation about the quantity of carbon that is stored, in fact, in this structure?
Do you mean carbon or do you mean calcium?
Well, can you answer to both? Yeah, it's not clear. Yeah.
Okay. I don't have an estimate on carbon content of in these inclusions or ACC, but I can say based on what the papers have been published and what they have reported about the calcium content in these inclusions. Mm-hmm. So, for example, if you take a model non-ACC forming cyanobacteria such as Synechocystis 6803. When you do a total calcium analysis on a sample, it would be about maximum about 1 milligram calcium per unit gram of dry biomass. And when you compare that to ACC forming cyanobacteria, depending on the strain, the values can range from 10 to up to 30 milligrams of calcium per gram of dried biomass. And that's a lot of calcium. And in fact, and the hypothesis is that most of that calcium is hosted in ACC. Mm-hmm. So that's something actually we some of the studies have explored that idea and into how this accumulation of calcium in such a high concentration could actually perturb the calcium geochemical cycle in these environments where cyanobacteria like forming ACC could be abundant. So it can be a lot compared to what we know in terms of accumulation of calcium in general in cyanobacteria.
Okay, great.
Yeah.
So there's a question from Jean-François. Jean-François, yeah.
Yes. Okay, thank you very much for the presentation first. Then I have two small questions. The first one is, I didn't understand if you get the infrared data in transmission or under a microscope and in reflection. Can you say more about this? And the second question is, since you have done some Raman spectroscopy, why did you choose not showing us some data? Okay. Can you tell us more about this? Thank you.
So first for the FTIR, so all the data which I've shown here, they were collected in ATR mode, so not transmission. And it was not done using a microscope. We simply dried our biomass and grounded it very finely and mounted it on a single crystal diamond IRE and measure the ATR reflectance spectra. And for us, it was the perfect choice because we were only interested in the carbonate region, which is the low wave number zone, and ATR is very effective in that region. For the Raman, when you mentioned, well, there's a very limited Raman data on cyanobacteria for the reason being that there's an autofluorescence issue with cyanobacteria. So the detectors on Raman get super saturated. So you don't really see any peaks unless you go on a different laser. So I don't have any data per se showing anything for that reason. I know people in the group in the past have tried some photo bleaching experiments to try to reduce the autofluorescence, but it's as far as I know, it was not very informative. Let me just say that.
Okay.
But
which wavelengths do you use for the Raman? Or have you tried?
If I'm not wrong, it's one of the wavelengths was 738, and the other one is yeah. So I have to double check on the values to be precise because it was a long time ago.
So you haven't tried the wavelengths close to 532 nanometer or?
No, I think we just have two wavelengths here, at least on the Raman we have in the lab. So the green and the red laser. So those are the two. And yeah. So
and with the green?
Yeah, I think with the green is what we have. At least I have tried. I don't know what other people in the previous members in the group tried.
Okay, so you cannot get any data close to 1087 wave numbers?
No, I didn't get any data on that. I know that at least for the cyanobacteria, it was completely saturated spectra when I looked at it for the 1080, which is more of a characteristic of carbonates. I know if you are more interested in the Raman data, I would direct you to the paper in geobiology on achromatium, because that has the entire Raman data on how you can detect ACC in achromatium, which will work very fine because there was not a lot of autofluorescence issues.
Mm-hmm. Okay.
Yeah.
Thank you.
You're welcome.
All right, so one more question, a lot of questions today from Amiel, please.
Hello, thank you for your presentation. I would like to ask, did you try to exploit the Overhauser effect to know the environment or the vicinity?
Sorry, what do you mean other effects in terms of
it's Noe. It's called Noe effect. It's Overhauser effect. It's in NMR. I don't know if in solid state NMR we can exploit that, but there is a kind of there is. I know there is a Noe technique that you could use, so you could know the local environment around the a molecule, around different different kind of yeah.
And this is a solid state NMR technique?
I don't know. Actually, I know that in NMR we could use it, but I don't know if in solid state it's possible. I don't know.
To the best of my recollection, we haven't used that effect. For our NMR analysis, which we did at LCMCP, we tried a lot of different sequences, which probes, which was mainly playing with the contact time and the to see how nicely we could separate the ACC peak, or at least see the interaction between phosphorus and carbon. But yeah, those were mostly based on what the literature studies have reported. So no, that would be my short answer because I'm not familiar with it.
Okay.
I think, sorry, I think you cannot to do some noise sequence with solid state anymore. It's not quantitative like this. I think so, but yeah, the team, the NMR team could answer, but I don't think it's the noise sequence is not possible by solid state NMR.
Thank you.
Thanks, Nadine.
You're welcome.
So, all right. So we have plenty of questions today. So, and yeah, it's almost time, I would say. So thanks a lot, Neha, for this very nice presentation and all this clear interaction and nice interaction. We wish you the best for your postdoc and for your future research.
Thank you, Lucy, for again the invitation, and it was really interesting to describe my work and get some feedback and a very interesting question and discussion. So thank you again.
Thanks. Well done. Goodbye, everyone.
Goodbye. Thank you.